So, the theme of my talk actually was on synthetic and natural molecules, but I picked CBD as an example for most of it, for most of my talk. So it essentially ends up being a thing on CBD. Um, so I compare what I call normal chemical reactions, which are inorganic molecules. And inorganic molecules all have some sort of an ionic interaction. They're like weak magnets. Okay, so if you take a look at, say, an oxygen and a hydrogen molecule and ethanol that's up there, the oxygen has a stronger pull for the electrons that are being shared by the two. So the oxygen pulls the hydrogen electron towards it. So oxygen tends to have what we call a slightly greater negative charge. And since the hydrogen's electrons are getting pulled away from it, the hydrogen ends up being a little bit of a positive charge. So, even a water molecule, the oxygen in the water molecule tends to pull the, molecules from the hydrogen towards it. So the oxygen in the water has a negative charge and the hydrogens have a positive charge. So in this particular example, you have, chloride ions that have a negative charge and they tend to, stick to the hydrogen that has a positive charge. And so you have all these like little magnetic interactions that are going on with inorganic molecules. However, I point out that organic molecules do not with a capital NOT behave like that at all. And there's that huge confusion because everybody thinks of chemical reactions and chemistry as the inorganic stuff. In the organic molecule case, especially when it comes to interactions in the human body, the molecule that's causing the ripple effect or causing things to happen are called ligands. Okay. And so, when you smell something, the perfume, that's the molecule that interacts with our nostrils. That's a ligand. That flavor molecule is a ligand. And based on what that smell is, receptors open up and you have this like sensation go to the brain. And that's how you say, okay, I smell this. I smell a skunk. I smell this sort of perfume. I smell a rose, etc. Each one gives a different sort of a sensation. So hormones, neurotransmitters, pheromones, all these things, right? They come in, and then there is some sort of a receptor, and this molecule that comes in, gives some sort of a signal to that receptor. And remember, this receptor is on the surface of a cell. And based on what this receptor on the surface of the cell is actually sensing, it has a trigger inside. There's a molecule A that's the tail of this receptor. And if molecule A is tail of the receptor, depending upon what this ligand actually stimulates in that receptor, it is going to sort of do something that triggers off a molecule B, and the molecule B is going to produce some signal that's going to start a chain reaction. So for example, I'm going to go back to the earlier slide. In case it's a cayenne pepper, you eat something hot, right? There is that, the cayenne, whatever, I forgot the molecule's name. It's not coming to my thing. Hits the tongue. Yeah, capsaicin hits the tongue, and the capsaicin basically, now it comes to, depending upon what happens, the sweat glands, right? There is one of the reactions that capsaicin has to that receptor. It's multiple receptors, is a triggering effect. And one of the triggers that happens, it tells the sweat glands to produce more sweat in order to cool itself off. That's one reaction. Another reaction is the capsaicin is going to trigger off, say, molecule A. Molecule A ends up triggering molecule B, triggers off a molecule C, and the molecule C goes and produces a sensation of heat, right? So sweating, sensation of heat, etc. So there are multiple triggers that are going on, multiple signals that are going off out of the reaction of one ligand to one receptor. And in my example that I gave you, it's different tissues, right? Different tissues are getting signals and different tissues are reacting differently. So what's very interesting about CBD, which is a cannabinoid, is traditional neurotransmitters that we know of are the things that the medical industry knows quite well: the glutamates, the acetylcholine, the dopamines, the gamma amino acid, butyric acid, the serotonins, endorphins, etc. This is what pharmaceutical drugs and pharmaceutical medication is based out of, all right? In all these cases, in this particular case, I have a receptor and the receptor is closed, right? The first diagram, it's an unbound receptor. In this case, it's closed. And then an endogenous receptor, right? Something that's inside the body comes in and touches that. In this particular case, it could be acetylcholine or dopamine, et cetera. When it touches that, the receptor actually opens. And in the case of a drug, right, the drug goes through the opening and then the receptor gets cleared off, the ligand gets cleared off and everything gets blocked off. So now all these things are what we call conventional neurotransmitters. Conventional neurotransmitters are typically the stuff that the pharmaceutical industry knows. Now, the cannabinoids are what I call a whole new class of unconventional neurotransmitters, okay? And they're unconventional because, well, it's unconventional because they're not classified by the pharmaceutical drug as a neurotransmitter, but they are, right? I mean, anandamide is known to be a neurotransmitter that our body produces, and anandamide actually works on the CB1 and CB2 receptors, right? These are endogenous cannabinoids that the body produces. But the cannabinoids that we consume are neurotransmitters, and they are both a neurotransmitter in the sense that they act as a ligand and do all these things, but they also act as a hormone. So they themselves act as a hormone. Instead of, acetylcholine is not a hormone. Acetylcholine is a neurotransmitter that triggers off something in that receptor. And one of that receptors in this, the diagram that I'm showing you, one of the trigger effects from that cell is going to be the production of the hormones. So traditional neurotransmitters actually do something to produce hormones. In the case of cannabinoids, they are both a neurotransmitter and they are a hormone in itself, okay? So in the cannabis plant, there are more than 80 cannabinoids. I mean, even the Kriya plant has got about 3 or 4 cannabinoids, but the cannabis plant has got quite a few cannabinoids. And the only two that we focus on is the THC and the CBD. Of course, people are now looking at CBG, CBN, etc. But there are about more than 80 to 100 molecules in the cannabinoid plant. And we know there are thousands of, receptor sites that these things work on. So the combination of just the cannabis plant, all the permutation and combinations of all these neurotransmitters on all these receptors we have in our body just goes in millions of combinations. So I'm trying to explain, this is one of the things I kind of indirectly did my PhD on. I didn't really understand it that well, but this is one of the most important receptors. Actually, it's not just the most important receptors, it's the largest class and the largest type of receptors in the human body. Most of the receptors in the human body are G protein coupled receptors, and they are in most of the critical functions that we do. So essentially, I'd like to simplify this. This is too complex a thing, but the G protein coupled receptors have, if you look at the black diagram on the right, sorry, you have a tail on the outside. This is the resting state, right? And there are 7 loops. If you look at it, there are 7 loops. All of them have 7 loops. And they have a tail on the outside where the ligand comes and interacts with. And then they have, if I'm not going to get too much into detail, but the head, which is at the bottom, has these, beta, gamma, alpha, kind of like, subunits. And those are the things that actually do a whole bunch of stuff within the cell. But essentially, you have a ligand, it comes from the outside, it interacts with the tail or the outside of this G protein, this receptor. And based on the ligand it is, that very same receptor is going to behave differently. And where it behaves differently is in the way the beta, gamma, and the what's that called, the G alpha unit, subunit. It's alpha, basically. It's the alpha, beta, gamma. So the way the alpha, beta, gamma subunits actually react and work, you have different things actually happening within the cell. So right? I kind of gave this point away. There are more than 80 cannabinoid ligands that we actually know, and there are more than 1,000 receptors in the human body. So if you do the permutation combination calculations of 80 receptors, 1,000 receptors on 80 ligands, it comes to more than 1 million combinations that in the human body. So and we're talking just one plant, right? I mean, what I'm building up is these are the number of ligands in one plant and the thousands of receptors we have on the body, on the complexity of the reactions that we get. So when we talk about just THC and we take just one receptor, the CB1, which is of course in the brain, and how the THC gongs us on the CB1 receptor, that's an extremely simplified way of looking at it. One receptor, one molecule, one reaction. The reality is that THC actually is going to be, having multiple different kinds of effects on multiple different kinds of receptors, and they're going to have multiple different effects on different organs in our human body. So does CBD. But interestingly enough, I'm going to send you another paper because it's something I wrote up. I did it like 6 years ago, and I don't think anybody understands it. I cataloged 23 or 24 different reactions going on inside the human body by CBD alone. Okay. And that's on a few different receptors that I've looked at. But that's a very complex calculation, right? So if you look at CBD and you look at the receptors that it affects the lungs, the spleen, the large intestine, the reproductive organs, the kidneys, the brains, the arteries. Most of these things I've cherry picked because the CBD has, and most of the cannabinoids have different types of effects on these organs. Now, why is CBD so important, right? If you look at the sympathetic and the parasympathetic, the CB2 receptor is very, very, very prevalent on the parasympathetic nerves. So these are the things that we don't actually control. We really don't have much control over these things. So our immune system, our digestive functions, our blood peristalsis, contraction and expansion, the heartbeat. So most of those organs have a dense collection of CB2, completely on an orthogonal thing. What is one element or one cell that has the highest concentration of CB2 receptors on its surface? It's a cancer cell. So if I take any cell, say a liver cell, a healthy liver cell, it does have CB2 receptors on it. But when it goes rogue and it becomes a cancer cell, it has about 1,000 times more CB2 receptors on the cell, right? And so that was a huge tip off for me right there, that when I see a cancer cell coat itself with so many more of the CB2 receptors, that's a tip off that cancer cell is in a very hostile environment, right? It knows it's going to get at any moment attacked. Slightest slip, it's going to get attacked by the immune system. And so it has that many CB2 receptors because that CB2 receptor is that critical for that cancer cell survival. Okay, and I won't stray from the point, but that density of CB2 receptors allows the cancer cell to detect a hostile environment, detect an attack, allows it to sort of like flood its surroundings with the checkpoint proteins to say, hey, I'm a friend. So it's that critical for a cancer cell, and it is pretty critical for a normal human cell. Now, a slight shift to the side, right? I mean, I am a huge fan of nature, and I'm a huge fan of using nature to heal itself, because natural products, and our DNA are very complementary. One of the simplest things that we humans try to do, or still do, is take vegetable oil, which is abundantly available and cheap, and try to simulate and make it into, animal fat or butter, which is far more expensive. And the only real difference between a vegetable oil and cow milk or cow fat is the fact that vegetable oil has double bonds, and which is, each bond has less hydrogen ions, and the animal fat has single bonds, which has got more hydrogen. So we took vegetable fat and we started hydrogenating it, bubbling hydrogen to it to saturate it. okay? And other than the flavor and the smell, you could get something that was saturated, except natural fats have cis bonds. The hydrogen bonds are straight. And the stuff that we did when we hydrogenated, we could never, even, that's one of the simplest, it's one of the simplest chemistry stuff you can do. And we still, after 80 years of doing it with all the sophisticated stuff we have, we cannot get it straight. It still twists, and it twists into what we call a trans bond. And why is that a big problem? Because our enzymes, our lipophilic enzymes that break down fats only recognize cis bonds. So all our enzymes break down the natural fats into the smaller subcomponents and everything is good. But the trans bonds, when it sees it, it says, I don't know what this is. It doesn't break it down. So you have this like larger aggregated clusters of this like mishmash floating around. And then when it gets into the bloodstream, it latches on to like the cell walls, etc. And that triglyceride unit or that trans fat unit, because it's like this ungainly thing, nucleates, that's one of the largest, reasons of arteriosclerosis or clogging of the arteries. Those trans fats also trigger the liver and the endocrine system into overreacting and producing more visceral fat, right? So it's a simple thing that we couldn't get right, but it actually caused a lot of damage in the human body for decades. And by the way, we've known it for 30, 40 years, and it took the FDA more than 40 years to ban trans fats in the United States. And even then, they gave the food industry 3 years to slowly ease it out, knowing well that it was actually clogging people's arteries, and it was the single biggest reason for heart attack deaths in the United States. So, not only can we not replicate a synthetic molecule perfectly into a natural molecule, our DNA and our bodies very clearly recognizes slight imperfections, right? Because a virus, that's how it tends to sort of like mutate slightly, etc. So the virus also has to mutate quite a lot, actually, because those little mutations it tries to do, our body's smart enough to say, no, I recognize you, right? So I recognize this change. So any change, even small changes to a natural molecule, the body has either body sort of totally ignores it and says, I don't recognize you, or it has lots of ways a negative reaction. So the best case for what I call a distorted molecule is you have a muted response where you're getting a positive response, but it's just in a lower form. That's actually good. The bad one is you get a distorted response where it's completely different from what you want, right? And so you think you're taking something that's good for you and your body's reacting negatively to it. And of course, the next, which is still acceptable, but it's a waste of money, is if your body just totally ignores it. So now you're taking something, supplements, you're paying money for it, your body is just ignoring it, you're pissing it out, and then you're paying a lot of money for it. So I think you've seen this before, but the pharmaceutical industry very normally and very commonly actually uses phytoceuticals. It uses plants. They follow the plant. They isolate plant just like we do, just like what Phytoceutical does. But in this particular case, the Visnaga plant has a compound called halin, which is a very, very good natural bronchodilator. And I can't patent kaolin. I mean, it's a natural plant molecule, right? So it's there, it's organic. Now, the pharmaceutical industry took kaolin and modified it and made it into a synthetic molecule called amiodarone. Now, amiodarone is actually quite potent, works very well, and is patented. So that's the active ingredient in most of these asthma inhalers that you actually have. It's amiodarone. But amiodarone is nothing but kaolin, right? Kaolin is not as effective as amiodarone, but amiodarone, remember, has side effects. Kaolin doesn't, and I think I point that out. So natural phytoceuticals, natural plant molecules have long history, right? It's been around for thousands of years. CBD, for example, has been in the cannabis plant for thousands of years. CBD has been in some of these hops plants for thousands of years. People have been using it, and it's absolutely safe. There's been no reaction from the body. So natural phytoceuticals have a consistent long-term history. and there's a long period of time, it's been dosed high, low, doesn't matter, it doesn't have a negative reaction. The efficacy of natural phytoceuticals is slower. It tends to sort of not work well. Rachel, we do have stuff that works almost immediately, but in general, phytoceuticals tend to take a little longer to permeate, saturate into the tissues, and whether it's a few days, a few weeks, it's usually 3 weeks to 7 days for the efficacy for it to start working. 1 month is a little too long. I would put that on the outside edge. Versus pharmaceutical drugs and synthetics, they actually have a very, very short historical, lifespan. I mean, 50 years, 80 years compared to 5,000, 10,000 years, it's nothing. All pharmaceutical drugs have tolerance, which means over time your body needs to have more and more of it to have the same effect, right? So that's and it also is quite addicting. Your body just gets used to it, and you have to have more and more of it. Even if you don't need it, your body still craves for it. Then a lot of times, a lot of people have rejection, right? Very high case of rejection. At some point when the body starts reacting to it, even when it didn't reject it before, it starts rejecting it now. Some people have instantaneous reactions. And that's why drug trials are very important. And so typically what happens is the curve at the bottom. It's highly effective in the beginning, and then slowly the effectiveness starts going down. That's the tolerance starting to build. And then there are less side effects, but pretty soon, in a matter of 3 weeks to 3 months, you start seeing the side effects starting to go up. And at the point where the side effects the problems of the side effects start getting to be more than the efficacy of the drug, you're supposed to stop it, right? There's no pharmaceutical drug is meant to be taken for more than 3 weeks, max 3 months. But that's one of the biggest problems we have in the United States. We use pharmaceutical drugs in a chronic fashion. It is a travesty. It's something that it's the American Medical Association looking the other way, the pharmaceutical drug industry looking the other way, and the food and the pharmaceutical industry fund 80% of the FDA's coffers. So, it's the most obvious, blatant, fact out there. Pharmaceutical drugs, absolutely, it's in the textbook, should not be used for any chronic period and for any chronic use, and it should not be used when the side effects actually start, getting obvious. Now, when you come to CBD, this is a natural, absolute sacrosanct fact. Any CBD that you get from the natural plant, now, of course, you can bastardize it and not extract it properly, but any CBD that you get from the natural plant has a double bond. I mean, it's isomerism, but most of the time that double bond is in that delta 2 position. That's the natural plant. However, when you overprocess the CBD, kill it, or when it's synthetically produced, you have it in the delta 1 position, the delta 6 position, the delta 5 position, the delta 4 position, the delta 3 positions. And I've shown the positions in every spot. So you'll have it in every position except the delta 2 position. And this reminds me very much of this trans fat example. That's why I gave that trans fat example, okay? That somehow in nature, and now our DNA picks it up. Our DNA and our immune system picks this up. So if you go look at the patent application I have for the antibody, Natium, the antibody for cannabidiol, we actually show the three-dimensional structure. We show which parts of the Natium, the antibody, actually touch which parts of the CBD molecule. We actually show the antibody antigen interaction in three-dimensional. We show the molecular latching. And this thing where the double bond shifts significantly affects one of the latches. So, for people who actually say, just monoclonal antibody, it's like, no, monoclonal antibody testing in every industry, it's there in pharmaceutical industry, it's there in everybody industry. Creating a pure monoclonal antibody, if you can isolate it for a molecule, it is the ultimate of tests. And I know that for CBD because even when people come, your GC mass spec, right, your certificate of analysis shows 99.96%, etc. Aha, what it doesn't show is this bond shifted out. It doesn't show it at all, right? It just, the peak comes out. That peak it's not sensitive enough to separate your peaks out. So if I take a CBD with a delta 2, a pure CBD, and then I take a one with delta 6, and I put them together, the GC mass spec is not going to separate it out. It's going to come out at the same peak. However, Natrium, the monoclonal antibody, which has been designed to latch on to the delta 2, when it's not on the delta 2 and the position is delta thing, it flips out. It doesn't latch. And so immediately that is, monoclonal antibody connection to the CBD is much weaker. You'll actually see less of them actually latching on. So now there are many different types of synthetic CBDs that are actually starting to come onto the market. And unfortunately, a lot of the scientists, well-known scientists that actually produce CBD are getting paid by third parties because it's a pain growing a plant. We know it. It's a pain growing a plant. It takes a lot of land. It costs a lot of money. It takes a lot of time. And then you get one yield in the case of hops. You get 3 yields in the case of hemp. And it's a hell of a lot easier to make synthetics, cannabinoids. It's much easier to make a synthetic CBD than grow it from a plant. So there are a whole bunch of what I call synthetic CBDs that are coming on to the market. And these are the common forms that you see. Quinolic groups are added, aldehyde groups are added, hydroxyl groups are added, fluorinated groups, alkyl groups, hydrogenated CBD, which is very equivalent to the trans fatty acids. By the way, that's one of the papers we published was hydrogenated CBD had been bought by some people in India for real cheap, some students. And we analyzed it and found it to be hydrogenated CBD. We told them not to take it. They still wanted to. So we said, okay, with the government of India, we did a trial. So it's a combination of, we did pure CBD, we did studied against hydrogenated CBD, and we studied against one of the cannabinoids that actually really got people sick. So we wrote a paper on it. And I think we're the only ones to actually have written a paper comparing all these different cannabinoids. So let me make sure that's the last thing. Yeah. So when you look at natural CBD in humans, right, and we published a paper on it, the liver is not stressed. In the case of ALT and AST, you actually find, compared to the control group, and I think this has been done over time over 6 months. The liver doesn't react much at all. And in the case of hydrogenated, here's your hydrogenated CBD, you actually see in a matter of 10 weeks, you see the ALT going up from 40 to about 450, right? On varies by person, but on the average 450. Now, the good news is at that point, when we stopped the treatment, when we stopped giving it to people, the liver recovered. And the same thing happened for the AST. So just that little thing, which is hydrogenating and a hydrogenated CBD, caused this to happen. So this is what happened when a synthetic cannabinoid, right? I don't know what the street name is, but it's most likely a synthetic cannabinoid called JWH18. The ALT levels, there were 3 men and 1 woman, but the thing about this was they were actually found on the beaches delirious. They were acting crazy. They were violent, etc. So they were having a very, very bad reaction to this. So you could see natural CBD had ALT, AST levels of about 40. The hydrogenated CBD, which did not cause that sort of drastic change in somebody's personality, had 480, versus JWH, which is this going crazy synthetic cannabinoid, knocked your ALT, which is very dangerous levels, 1,400, 1,200, very, very high. And in some cases, this actually caused permanent liver damage also. So we know for a fact, and there are so many, because most of these synthetic CBD reactions, people end up in the emergency rooms. There's more and more evidence and more papers being published on the dangers of synthetic cannabinoids. But these are what I call THC mimetics, right? So it kind of gets your brains nuts and makes you go cuckoo. However, it's not far to imagine that if you're going to get into synthetic CBD, your body's not going to respond the same way. A cannabinoid is a cannabinoid. So if you can see, the only good thing about a THC and these mimetics of THC is you get instant feedback, right? Because it messes with your brain. So you have this instant neurotic or get high. There's that instant feedback. CBD does not give you that feedback, right? CBD is a much more innate work within your body kind of molecule. However, when we look at hydrogenated CBD, you could see a tenfold increase in the liver enzymes in the stressed out. So the liver was getting stressed out. So that's the only way to really see whether something is natural or not. So it's kind of really interesting because the only approved CBD by the FDA, right, Epidiolex from GW Pharma, it has in fact, we probably shouldn't write it. Somebody will figure it out. If we put a paper out without mentioning it, somebody will figure it out, Brett. But it has a profile extremely similar to the hydrogenated CBD, so synthetic CBD. It does not follow the profile of a natural CBD, right? So in the trials, they caused almost every, it wasn't a large trial. It was just a trial starting off with 75 in the control and 75 in the actual trial group, which is not a large trial, and few of them fell sick and went off, and very few finished the whole trial. But pretty much the study said that almost everybody who was in that trial, every one of the 75 had a severe reaction or a moderately negative reaction, which you don't see from a regular CBD. So the battle that's happening in the future now are natural cannabinoids, including natural CBD players, squaring off against synthetic CBD players. And what the synthetic CBD players are playing off is this whole information source from natural CBD saying it's safe. So this reminds me again of all the papers and everything that's been written up from the 1960s that says, cannabis is safe. But the cannabis that they're talking about has 1% THC. That's what is naturally there. If you look at the cannabis plant that's in the dispensaries now, it doesn't look like anything like the cannabis plant in the wild. The cannabis plant in the wild is tall, very scrubby, has small flowers and 1% THC. These plants are very small, have huge inflorescent flowers, and they have like 30% THC, 28% THC. Right, and they don't look like, they stink. They smell like crazy, but they don't look like a natural plant. However, if you leave these plants be, remember they're asexual, they cannot breed. But if you let them be, they actually lose their potency. They actually, if you sort of like don't crossbreed them and you don't do tissue culture and you let them be, they'll just die out because they don't produce seeds. Or if they do germinate and produce seeds, the THC levels drops drastically and goes back to nature and regresses back to nature. So everybody uses these safe data points and safe molecules and safe studies to come up with a new molecule. And then there's an assumption that these new molecules are just as safe as the parent molecule that they're trying to mimic. But this is why I go back to humans and our efforts in messing with nature. We have never succeeded, by the way. We have not succeeded with a simple thing as hydrogenating fat. We've not succeeded in trying to make a synthetic out of THC. And I can guarantee we're not going to succeed by making a synthetic out of CBD. You can make a synthetic out of CBD, but it's not going to react the same way in the human body. And most likely it's going to have a negative reaction. So this is not directly to our point, but a lot of the vapes, even CBD vapes, were causing a lot of problems, and so it was the additives in the vapes that were actually causing problems. But as an aside point, I have not created a vape product, although there's been a lot of demand for the vapes. And a few years ago, people are like pounding on our doors for vape. But when we looked at the temperature, there were no cold vapes. Everything was heated up. The temperature in the vape pen to heat up the CBD denatured. It's called pyrolysis. And it's a scientific word called pyrolysis, which is heat denaturation. Denaturation. And we found that that temperature denatured the CBD, and I didn't have enough CBD to go around. We needed the CBD for people who truly needed it. And so I just refused to give CBD that somebody could, they would pay us for it. We'd make money, but I just couldn't see it getting heated and destroyed. I think I made this point before. Um, so all our analysis that are done out there is GC mass spec, and I've quoted the science. This is not a paper written by us, but it's a paper written by somebody else. But it's a well-known fact that the minor isomerisms and the minor changes in CBD cannot be detected, and even some of the synthetic CBDs cannot be detected by GC mass spec. They'll show a peak in the same spot as pure regular THC. So the lab guy who's doing the work, he cannot detect that this is not CBD, right? There should be a shift where they say, okay, this is not a CBD peak, but you don't actually see it. That's why the GC mass spec things are quick and dirty methods. It's fine, but I'm not a big fan of it for truly measuring the quality of CBD. Quality, not quantity. So X-ray crystallography is actually very precise. It is a three-dimensional model. It actually shows all bond angles. And if one bond angle, this is a great way to see the isomerism shift. If it's not on delta 2, somewhere else you can see it. But there are a handful of these machines in the world. They're very expensive to use. And even good labs don't have access to an X-ray crystallography machine. UCSF has one, but a very good lab like Anresco doesn't have one. It's too expensive. They're too finicky. They need to be refined. This is like a very high-end scientific equipment. So we have a patent pending for the monoclonal antibody test. And so what you do is you take CBD and you inject it into, we did rats, mice, and rabbits. And so there's an antibody that's actually, there is not one, but there's a whole bunch of antibodies, right? It's polyclonal antibodies that get produced to that CBD. So you have to go and you differentiate it and you pull out all these different antibodies and then you isolate and you find out the one that has the highest clumping effect, the strongest, reaction to the CBD. And then you turn it and you make what's known as a hybridoma. You take that particular thing, you put in the yeasts, and you start now producing. It's like growing yogurt. You get your pure hybridoma with that one monoclonal antibody that you want, and then you start like producing it like crazy, right? And so then we call that, like I call the Kriya plant, the Humulus unanensis variant that gives CBD. I had the right to name the plant. I called it the Kriya plant. I had the right to name this molecule. I called it Natyam, which in Sanskrit means dancing. So I call the molecule Natyam. And, again, we have all the very, very exactly which molecule or which part of this Natyam touches which molecule of CBD and which angle and what changes and what you see here and there. We've got it all documented. It's all part of the patent. And if you look at the measuring of the thing, right? So you take a CBD and you take a look at any concentration of that CBD. If it is 100%, if it's totally bioactive, it's 100% bioactive, we expect a certain amount of Natyam to be bound to it, right? We know that. We know what that perfect thing should be. But on the other hand, when you actually take a look at binding, you know the amount of CBD you have, and you put the Natyam and you take a look at the complex that's actually formed. And if you find that the complex, and I'm showing the actual reading as in this case, green, right? So if you take a look at the green, you take a look at the measure of the green, and if your expected reading is 100 and your actual reading is, uh, yeah, your expected reading is 88 and your actual reading is 20, then your bioactivity ends up being 22%, right? It's a pretty straightforward calculation. But the trick is to actually having that pure monoclonal antibody. So we've looked at CBD from the inflorescence, we've looked at it from the shoot tip, we looked at it from the leaves, we looked at it from the bark, and we've actually found the inflorescence to have the highest bioactivity, absolutely. But you can actually see when we see bioactivity that's very, very low in the 8.08, 0.09, etc., it's fake, it's simulated CBD. So this is hemp. The stuff at the bottom that you actually see is, I should have shown a bar, but at the hop CBD, the lowest we have is 0.78. We use that for pets, and humans normally we have 0.92, 93, 95. So we're in the 0.78 to 9.98 range. And this is the commercial stuff. The commercial low that we've seen is about 18, and the high is about 40, and the average is about 0.28, 0.29 in hemp. So what do I mean by CBD quality? So natural CBD is actually the healthiest. It's easier on the human body than synthetic CBD. It doesn't have any negative reactions. And this is one of the biggest worries I have. There's more and more synthetic CBD coming out in the market today. The only thing that's approved by the FDA, Epidiolex, shows all traces, all profiles of a synthetic CBD. And so what people actually want, what people are looking for, what people, write about and say this has been a very healthy thing and cured me this way is naturally is natural CBD. The other big point is vertically integrated vendors have far more control over the quality process. What do I mean by that? When somebody is growing the plant, when somebody has the seeds, grows the plant, does the oil extraction from the oil, does the isolate extraction, all that stuff, they have tremendous control over the quality. Now, there was a guy called Randy Kindred and he's one of the largest hemp CBD vendors in the United States. He came to us years ago and said he wanted to improve the quality of his CBD, and he was getting hemp CBD. And he started sending us stuff to test, and we were finding it to be, normal CBD thing, but he wanted to improve it. And then when he went down and I said, okay, this is what causes it. And this is what he couldn't go down the chain because he was at the isolate manufacturing thing, and they had no idea where the oil was coming from, and it was totally blended. So they had no idea who to go to talk about how did you make the oil. And so when we did go down to oil guys, they were getting these boatloads of like plant material show up. And it was all mixed. They had no idea whose plant, what was there, because you had these brokers who were buying these things and sending it to them. And when you, of course, went down to the farm and talked to the people who were farming, we could have good conversations, said, you need to do this, you need to do that. But that's all in the growing part. But then they had no idea where their plants were going to get processed. So this is the problem this is the problem with the non-hops CBD, with the hemp CBD. You have no vertically integrated players. The whole industry has become these, silos where you have farms, and farms just get paid a lot of money for people to just come and collect the stuff and take it down to a processor who has no idea. There's a middleman who's taking it and taking it to the processor. So by the time it goes down 4 chains, there are 6 different elements in the middle. And I think that holds the industry back a lot. So X-ray crystallography is one of the best ways to detect natural and synthetic CBD. It's too expensive, you can't do it. Bioactivity test is one of the critical things. We don't make money off it. We use it for our own testing. And we have the smarts to come up with a patent and the monoclonal antibody. If there is and we have actually licensed it out to a third-party lab to test it. But again, there's not that much money in testing. But If you really want to get your product tested, the bioactivity test or the extracrystallography test are the best ways to do it. Now, natural CBD does not stress the liver. I mean, we've had people be taking it for years now. Our product 5 years now, and when they've gone and done their liver tests like 3 months ago, still no stress. Simulated CBD, right? Synthetic CBD drastically elevates your ALT, AST levels. Within 10 weeks, you'll actually see the big difference. And in a nutshell, of course, I talked about GC mass spec and the common labs not measuring quality, but measuring quantity, and not being able to differentiate between even small changes in the molecules. Even major changes, it cannot differentiate it. I think that's it.